Thank you very much for the invitation. It's my first trip to Texas A&M University. I've been to Austin and Weslaco, been to some places in Texas, Dallas, but I haven't been here to the university, and it's been a really good day so far talking to students and faculty. This afternoon I'm going to talk about emerging plant diseases and their threat to global food security. I'll discuss many of the things that Fred just mentioned in my bio. If you read the 2012 PCAST report, the President's Council of Advisors on Science and Technology, the U.S. is the undisputed world leader in ag production today, but as we look across the 21st century, we face a lot of challenges, including managing new pests, pathogens, and invasive plants, increasing the efficiency of water use, reducing the environmental footprint of agriculture, growing food in a changing climate, managing the production of bioenergy, producing safe and nutritious food, and assisting with global food security and while managing abundant yields. So new pests and pathogens, invasive plant diseases are having an environmental footprint. If you look at some of the pathogens that require a lot of pesticide use for management, and they're affecting food security in a lot of the developing world. If you look at human population growth, we know we've got more than 7 billion people. Today it's going to reach 8 to 10 billion people. 60% of the people in the developing world lack basic sanitation. 1/3 have no access to clean water. 1 billion people are illiterate, and 2/3 of them are women. So the education of women is very important in moving toward a more food secure world. And 15 million babies are born to women between 15 and 19, and there's been a clear evidence showing that when women are more educated, they have smaller families, their income rises. So we have a lot of issues besides plant pathology or horticulture to deal with in managing food security, but some of this involves education. I'm going to end my talk talking about capacity development and education. We need to produce food sustainably, increase production of nutritious foods, raise yields to smaller income, smallholder farmers, conserve land and water, improve health through accessible foods, adapt to climate change, reduce environmental impact, and reduce food waste along the value chain. And there's been a number of glossy publications from the Chicago Council in recent years about this kind of agenda for sustainable intensification of agriculture. Food security is tightly linked to national security, and if you look at this graph, this shows the number of riots and unstable conditions in many areas in the developing world, some in the Middle East, some in Africa. And these riots kind of coincide with food price spikes. So oftentimes you'll see countries becoming insecure when their food supply is insecure. And Secretary Kerry, a quote here, "A lack of access to food leads to malnutrition, instability, even violence. There's been food riots in different parts of the world. And development assistance is much cheaper than military assistance." The State Department has the 3 Ds, development, diplomacy, and defense. And those 3 Ds are kind of all interworked together when you're dealing with food security. And plant pathology is right in the middle of this because plant pathogens have caused food insecurity at many points in time in our past. If you look at the late blight outbreaks of the 1840s in Ireland, when this Irish family uncovered their potato tubers that were rotting in the field, the Irish people were totally dependent on monoculture of a single host plant, and they lost their food. Many people died. 2 million died. Many immigrated to the U.S. There are statues like this: "Never again should a people starve in a world of plenty." This is in Harvard Square in Cambridge, Massachusetts. This is a mass burial ground in Canada, Grosse Isle, a quarantine ground site where Irish immigrants were quarantined before they entered the United States. We didn't know the germ theory in those days. We didn't realize that pathogens, even human pathogens, could cause disease. People were packed into these ships, the coffin ships, and they wound up dying from human disease because of a plant disease outbreak. We also, in the past, have had issues with famine in other areas of the world. Brown spot of rice caused the Bengal famine in the 1940s. 4 million people starved from this famine, and there were political decisions made at that time to move food to feed soldiers instead of people that were suffering. So sometimes political decisions happen, the bigger view of winning the war over dealing with starvation in parts of the developing world. And this was the Bengal famine caused this famine. outbreak. And then we have Uganda in the 1990s, cassava mosaic virus, which just kind of has expanded its range over the country. They have 30,000 hectares down to 3,000 hectares. Starvation, political instability, you have girls sold into slavery or sold from their families, early marriage because of a cassava virus outbreak. And then coffee rust in 2013. There were states of and actually 2012, 2013, states of emergencies called in Central America. 400,000 workers lost wages due to loss of a crop to harvest. Basically, these are defoliated coffee plants on plantations in Guatemala. These are the lesions caused by coffee rust. In the past, this disease has not been a major problem. It's been managed with fungicides in many areas or not an issue at high altitudes, but it's become a crisis in recent years. So there are a lot of armed and dangerous plant pathogens, and if you look at this 2010 science report, you know, they named stem rust, potato blight, sigatoka, rice blast. This is aspergillus on peanut and corn. So why do we have these diseases emerging? Well, we've got pathogens that are changing their geographic range. They're moving with trade. They're changing and evolving in pathogenesis, or they're even newly evolved, never been described before. So there's lots of reasons why we have emerging diseases. I want to go through a few of these. This cassava mosaic virus and cassava brown streak are a major part of the AID and the Gates Foundation research portfolio. And these diseases are causing serious problems for cassava growers and people that eat cassava in Africa. The pathogen actually jumped host in South America. It wasn't a big problem, but when cassava was introduced into Africa it found this accidental host, they think the virus actually might have moved in from weed species, and they're not really sure how it moved in, but it's caused $60 million worth of losses, and it's a huge problem. And the stem rust pathogen, UG99, another example where it's kind of discovered in Uganda in 1999, and it's migrating into the Middle East and into India, and this is a serious threat. There's been a lot of new breeding work done to control this strain of stem rust, and so when you have new outbreaks of new pathogens, new lineages of pathogens, it can cause food crises. Black sigatoka is a major problem in Central America. Cavendish bananas are grown widespread through Central America. One type of Cavendish bananas, and Dole and Chiquita spray over 70 fungicide applications. I have 50 here, but it's actually higher than that. And they use aerial airplane applications. They have fungicide-resistant strains. So sigatoka is a big problem. It's expensive to control, and you know, they need to diversify production systems. And we're doing some studies to understand the population structure. And you know, potentially transgenic bananas may be a solution. We may need to diversify just the types of varieties, because Cavendish is also very susceptible to Fusarium Panama disease, and that's looming on the horizon, not in the Americas yet. I want to take a few minutes to talk about coffee rust, because I know there's folks here at Texas A&M that are interested in coffee rust. The World Coffee Research Foundation is now here, funded through AID. Coffee rust was not a major problem until, you know, it's kind of been a background looming pathogen. This is a picture of a lesion, a set of lesions I took on a coffee leaf in Guatemala. These lesions are huge on the coffee leaves. Usually you see these smaller discrete lesions. We don't know if there's a new race of the pathogen. We don't know if a new genotype is immigrated, but we know there's a lack of resistant varieties. There's more disease at high altitudes. And the shade-grown growers at high altitudes have not sprayed for coffee rust in recent years, and the plantations are very old. Plant is a very susceptible variety. So this is a slide I borrowed from Jacques Avelino, who's at Cartier, a CIRAD scientist in Costa Rica, and climate change may be involved in coffee rust. If you look at the, I'll back up, I went through that too fast. El Nino effects may be affecting the incidence of coffee rust. You get higher temperatures and a longer, the latent period is shortened, and the disease occurs at higher altitudes with low rainfall, but enough for germination. You'll see dispersal being increased. These dried spores are moved by the harvesters and workers. And since coffee prices have been low in recent years, they haven't been able to apply fertilizers or fungicides when needed. So the preventative controls were not in place. So there's just a series of things affecting these outbreaks, and it's not always just the new pathogen immigrating, but it's policy in the countries, the lack of science development at the coffee institutes, the lack of plant pathologists in the region, very few plant pathologists. So I encourage you guys to go work, the students to go work in some of these areas where these emerging diseases are problems. And what they're doing now is replanting stumping plantations, replanting. Interestingly, climate may be involved in increasing this disease, but with large areas defoliated, there's a reduced carbon sequestration over large areas of Central America. So the coffee rust itself may be having an impact on climate. So while I was at AID, I worked on a number of actions to help solve this problem, and integrated disease management strategies, loan packages for ramping up planting material, the pre-breeding program, and World Coffee Research, Tim Schilling and Dr. Lombardini's work here at Texas A&M, coordination units within the coffee institutes, developing rust-resistant varieties. Also, this need to test Ethiopian germplasm, which may have some resistance, but there's been issues with getting release of this germplasm. And there's also needed research on the pathogen end, not just the breeding end, but developing rust monitoring systems, sampling rust populations, determining what races and whether there are new genotypes or lineages, and then just training the next generation of coffee researchers for Central America. So these are all things I worked on as a Jefferson Science Fellow while at AID. So there's lots of drivers of emerging plant diseases. I mentioned climate change, also trade, global trade, movement of plant materials. Pathogens move in seed, they move in transplant material, in tubers. They move when people move their favorite host plant with them and conceal them when they're going through the airports. Pathogens do move with trade. And we know in developing countries there's a lot of non-traditional crops being grown, and sometimes just agriculture change itself can introduce new pathogens or ones that jump hosts that we hadn't seen previously. And the fact that we have only 4 major staple food crops and lots of just very limited genetic diversity in many of these crops, there's an interest now in just sequencing genomes of orphan crops, especially for areas in Africa where we can potentially change the diversity of foods that are eaten. And then I mentioned host pathogen jumps. So that was just kind of an introduction to emerging infectious plant diseases and issues that we're dealing with now and we have in the past. So I've spent most of my research years working on phytophthora diseases, and I work on light blight, which is considered a threat to food security. The disease has definitely increased in incidence and geographic range, and we're finding it on different host species. It's occurred on petunias, flowers. It moves into other hosts other than just potato and tomato. This is a grower's field in Honduras, and his potatoes were pretty devastated by late blight, and he has limited access to fungicides. So basically, he just harvests what he can from this field, and then he saves seed, which may be infected, and kind of continues this cycle of disease. Late blight is considered a re-emerging disease. We know it has an airborne phase where the sporangia can travel over kilometers, but the pathogen mostly moves in plant material, in tomato transplants, or in tubers. And we know it's a polycyclic disease. There's many stages of spore production within a growing season, and these spores move into non-infected plants. We know there's some lineages that are resistant to fungicides. The pathogen can shift hosts, and the genome of the pathogen has been sequenced. We know it's a plastic genome. There's a lot of effectors, and these effectors can overcome host R genes that have been deployed in some areas of the world. But mostly we have, in the U.S., susceptible potato varieties being grown, and most of the tomato varieties are susceptible as well. In 2009, we had the worst late blight outbreaks on potato and tomato than we've seen since the 1990s, and the culprit was tomato transplants from the Bonnie Corporation that were shipped to the Northeast from one source, and these transplants were infected. If you see, there's some lesions here and lesions. Homeowners bought the plants, they planted them in their backyard, didn't spray the tomatoes died, and a lot of the headlines in the New York Times and in Science News was, we're having a tomato disaster. What's happened? Well, there was one source, and the source, we identified the lineage. It was the US-22 strain, and it was a different lineage than we'd seen before. And so a bunch of us mobilized, extension research faculty. We wrote a CAP grant to USDA NIFA, and we were funded. Howard Jefferson at UC Riverside was the lead PI, but there were lots of subawards to different faculty at Cornell and my lab at NC State. And our lab developed a real-time disease tracking and alert system for late blight. And we had some other prototypes, the cucurbit downy mildew and blue mold, tobacco blue mold alert and tracking system. So we used that as a model, developed the usablight.org tracking system. And growers can report in where the disease is. They can get disease alerts on their cell phone, and we can determine if the pathogen's in the area, and that's a cue for them to start spraying a fungicide if needed. And we also have a decision support system that allows growers to determine whether weather conditions are conducive, because just because the disease is in the area and the pathogen there doesn't mean that weather could be conducive. Very high temperatures, dry conditions, you won't get spread of late blight, but high humidity and rainy conditions you will. So we have a weather-based forecasting system also embedded in USA Blight that was developed by Bill Fry at Cornell. And then we're also generating information about the genotypes and the phenotypes associated with the various genotypes of P. infestans. This is just what the disease alert and forecasting system looks like in 2014. Usually our outbreaks first occur in Florida. Florida has winter tomato production. Also Mexico has a lot of the Florida growers grow in Mexico. So there's winter tomatoes here as well. Right now it's just USA blight, but we're planning on extending this into Central America and Latin America and starting a Latin blight network. But the disease usually occurs here in the winter, and then you start seeing outbreaks in the Northeast, very concentrated. Maine is a big sweet potato producer, lots of late blight in that area. Western North Carolina, we grow state tomatoes. We have different lineages in the western part of the state than the eastern part of the state where our potatoes are grown. And there are tomato repackers that take fruit in Florida, they repack it, and they ship it north. We've got a major repacker in western North Carolina, and culled tomato fruit can also be sources of inoculum. So we're starting to connect how this disease moves with not only tomato transplants, but ripe tomato fruit. There's been this trend to grow ripe tomatoes and ship ripe tomatoes and not green tomatoes. And these ripe tomatoes, if they have late blight and they're dumped outside a packing shed, they can sporulate and cause disease in neighboring areas. So the USA Blight system's been up and running for 4 years, and we have lots of data now in the system. We're doing microsatellite genotyping, so we can tell what lineage quickly using a 12-plex microsat assay, and then we're assigning phenotypes, basically fungicide sensitivity associated with the different lineages. In 2009, we missed the opportunity to use the fungicide mefenoxam to control late blight. We did not know it was sensitive to mefenoxam because the lineage previously that was common, US8, was very resistant to that fungicide. So now we know the fungicide sensitivity of many of the lineages, and we've been able to deploy this decision support system. And Howard Jefferson's been working on a rapid assay for detection of fungicide sensitivity. The folks at Syngenta and Scottish Crops Research Institute came out with a marker associated with RNA polymerase I, and that there's a mutation in that RNA polymerase I that may be associated with mefenoxam insensitive strains. So we've been looking at that as well. So we've got real-time diagnosis and tracking. And I just wanted to show you this change in lineages. US8 was this mefenoxam-resistant lineage, and US11 is resistant. But you see this expansion of US23, which is also sensitive, and it goes to both. This is potato and this is tomato. This US22 lineage was really mostly on tomato, and it's pretty much disappeared now, and we see mostly this new lineage. We don't really understand fully why we're getting this displacement, but we know we get introductions of new lineages each year, and usually the source is from south in Florida. There's a lot of importation of materials into Florida as well. Tomatoes coming from Thailand and Taiwan, Southeast Asia. The US11 lineage is really common in Taiwan, and it's kind of reappeared just in recent years. So there's the movement of planting materials is having an impact on what genotypes we see. We've looked at the microsat the 12-plex microsat loci, and the colored lineages are the most recent lineages. This US1 lineage was the putative ancestral lineage thought to have caused the Irish potato famine, and we don't see that lineage anymore in the U.S. But this is the US-23. It's a close relative of US1, but distinct based on the microsat and whole genome sequencing. So we've got about 6 different clusters of groups of isolates. Fungicides are expensive, and growers, by using this decision support system, they can target when they spray, and we have an opportunity to improve farm efficiency and fungicide use. If growers can reduce their applications, they can save a lot of money. So that's been part of the justification for using USA Blight. And now we know US8 and US11 are highly resistant to Methanox, and the other common lineages that are in the US are really sensitive, and they don't grow at the doses you might find in the field. So we now know the growers can go back to using methanoxam, but they also need to rotate with other modes of action so they don't develop resistance again. So this is kind of a little conclusion section on, we've got the shift in lineages, we've got some methanoxam resistant lineages, some are sensitive. We also tested a whole range of other fungicides that are targeted for oomycetes, and none of the lineages were resistant to those other fungicides. So that's a good thing, and that means growers can also rotate with these other compounds. I want to switch gears a bit and talk about historic late blight outbreaks. This has been a big interest of mine. It was proposed that the US1 lineage caused the potato famine, and that this lineage, the source of the lineage was from Mexico. But back in the 1840s, the first reports, this is the diagrams from Austin Bork's Nature paper in 1964. The first reports, just based on reading old records, were around the ports of Philadelphia and New York. This is in 1843. The famine outbreaks in Europe did not occur on a wide scale until 1845, 2 years later. So you had first epidemics in the U.S., 2 years later disease in Europe. And the disease outbreaks were around the ports. So that tells you maybe something was shipped in through the ports. And when you go back and look at the ship records, you can see that the ports were shipping in Peruvian bat guano, and they were shipping in potatoes from Peru and South America. So my hypothesis was this thing really, the outbreak that caused the famine came from South America and not from Mexico, which is the Mexican theory of the origin really kind of developed in the early 20th century when Reddick and John Niederhauser did work for the Rockefeller Foundation in Mexico, and they found both mating types of the pathogen in Mexico. They found Solanum demissum, which had some resistance genes, and then over years they started finding more genetic diversity in Mexico. So the idea that Mexico was the center of origin and source of outbreaks is really a 20th century idea, but the early scientists believed that the outbreaks really came from South America. And the historic literature suggests that as well, although we didn't know in 1845 that pathogens could cause disease, but there were anecdotal records of the Indians knowing of this disease, cutting the tubers, throwing out the diseased parts, and planting the healthy parts. So there was already some cultural practices in place to manage this disease, and there was lots of diversity in the Andean region, so late blight did not take off like it would in a monoculture in a country like Ireland with one variety. So, we went out to study, what's the source of inoculum and what strain caused the famine. Did this US1 lineage that Cornell group, Bill Fry's group, found it was very dominant in the mid-20th century. So we asked the question first, did the US1 lineage cause the famine? And I did this by going back into historic herbarium specimens from actual outbreaks from the 1840s. I was at a meeting for the 150th anniversary of Phytophthora in Ireland, and I met Mary Robinson, the president of Ireland. It was a big hoopla, big meeting, nice meeting in Ireland. And on my way over, I stopped at Kew, and I found all these samples. So I started digging into these things way back in the late 1990s. And in those days, we could not do whole genome sequencing in those days. We didn't have a Sanger sequence genome. All we could do was PCR, RFLP. We could look at mitochondrial lineages. So I had a series of postdocs that worked with me. And we went in and we looked at mutations in single SNPs in the mitochondrial genome that would sort out the 1B lineage from the other lineages. And there was 3 different regions in the mitochondrial genome. This is just showing where one of these MSP1 restriction sites were. We went through about 200 samples just doing this SNP analysis, and everything was not the 1D, it was the 1A mitochondrial lineage. So we published this paper saying we don't think the US1D mitochondrial lineage causes famine, we think it's a different lineage. And then I had another student, a Fulbright scholar from Costa Rica, Luis Gomez. We went back through a whole series of collections of modern isolates from Bolivia, Brazil, Peru, Ecuador, Costa Rica, Mexico, US, Ireland. And we sequenced both mitochondrial and nuclear genes using multi-locus genotyping. And the ancestral mutations, the oldest mutations, were always in Andean samples. They were of South American origin. And Luis also found this highly divergent lineage that we subsequently, with the folks from FIP, named Phytophthora andina. And Phytophthora andina is only found in Ecuador, Peru, and Colombia. And this was clearly divergent from all of these modern Phytophthora infestans lineages. It had lots of mutations, and it shared a common ancestor with Phytophthora infestans. And we found that there was only one of the 2 major lineages in Mexico, only this lineage, not the other, and more shared lineages between the South American and Irish samples than the Mexican and Irish samples. So we published in PNAS that we think the South American origin for this disease. Well, time kind of moved on. A big group of us got together, and with the Broad Institute, we did the first Sanger sequence of Phytophthora infestans from a modern lineage from the Netherlands. And there was a nice paper in Nature. It took $2 million. I mean, it took a lot of money to do this first Sanger sequence of this genome. But now we're doing Illumina sequencing. We still had samples from the original outbreaks from, this is from Europe, from the outbreaks in the European side in 1845. So I started a collaboration with a postdoc, Mike Martin, who's now at UC Berkeley, and Tom Gilbert. And Tom Gilbert runs the Center for Geogenetics at the University of Copenhagen. We extracted DNA from a series of modern lineages, and my lab sequenced these in North Carolina, US 82223, and then Tom's lab sequenced the 19th century genome shown in red. And we saw, there was definitely differences between the modern lineages. European historic samples, even between 1840 and 1880, were divergent, suggesting that maybe over that 20-30 year period there had been more than one introduction of the pathogen. Because the theory was there was a single introduction of an A1 lineage from Mexico that predominated for 50 years. That was the original theory proposed from Cornell. And I suggest that maybe there were multiple introductions. We see that today, and that these lineages may have been displaced over time. So we've now done next generation sequencing of about 50 different historic outbreak samples. These are mitochondrial genealogies. We published in Molecular Biology and Evolution. Samples, at the same time, 2 years ago, Sophie and Tamun's lab in the UK also sequenced some mitochondrial genomes from P. infestans, and they suggested, once again, a single lineage. They're naming it, it's a 1A lineage, but it's the ERB1 lineage. And so then they also suggested it went extinct. So we went back through all these samples, and we found that this lineage is not extinct, that the famine era lineages, these mitochondrial lineages, are present in the Andean region, and we also found a couple samples from Mexico. So ERB1 is not extinct, but once again, the most divergent lineages, the oldest lineages, are of South American origin. These are several other mitochondrial lineages, and they're all from South America. So there's more diversity in mitochondrial genealogies from South American lineages. And then this paper is in review right now in Molecular Biology and Evolution. But what we've now done is sequence. This is mitochondrial genealogy, but this is the Illumina genealogy from the whole next-gen sequencing. And we found that this ancestral Andean lineage, P. andina, shares a lot of admixture with the famine era outbreak lineages. So once again, the Andean lineage is ancestral. This is a maximum likelihood tree, and this is the US1 lineage. It emerged later. These are the Mexican lineages from Toluca. They emerged later. And these are modern aggressive isolates like US-22, 23 emerging later. And then a bunch of samples from South America that are more recent. So this pathogen is not only, it moved up into the US, moved over to Ireland during the famine, but this pathogen also has moved back into the Andean region because potato germplasm is going back into the Andes. It's moved out of the Andes, and then people are testing germplasm, and there's movement of Phytophthora back into the Andean region. So that's why this is more recent. These recent lineages from Peru are more recent. The other really interesting thing that we found is we phased the haplotypes. And, the theory that Mexico is the center of origin was also based on the fact that the pathogen reproduces sexually there. So when you phase the haplotypes of these lineages, we see that the P. andina lineage, the famine era lineages, these are phased haplotypes. They're clonal. So this pathogen wasn't reproducing sexually, and it still doesn't reproduce sexually in the Andean region, but in Mexico it does, and that's because both mating types were moved into Mexico. In the Andean region, the mating types are isolated geographically. There's a zone between Peru and Bolivia, and these mating types are on different hosts in different parts of that country. But when they were moved into Mexico, probably by indigenous people migrating up, they brought this pathogen, and it's reproducing sexually there now. And interestingly, these stars, the basal lineages of all the Mexican sexually reproducing populations, are all on wild Solanum hosts. So that was a really interesting discovery too. We've got Solanum demissum, Solanum stolonifera, Solanum Colombia, I can't remember the third species, but they're all wild lineages. The pathogen was moved by indigenous people on these wild Solanum hosts. So this is kind of hot off the press. Well, it's not even on the press yet, but it's been submitted for publication. And I think it kind of explains a lot about why, even when you go back and read John Niederhauser's reports in the 1950s about late blight in Mexico, when he found it on Solanum demissum in some isolated areas, and it still occurs there. So we're also kind of now doing, since we can do microsats quickly and cheaply, we're going through lots of herbarium samples. This is just some samples from over the last 150 years. You see Herb1 lineage kind of peaks at different times when you look at the herbarium record, but this is the US1. So it's kind of, we're seeing a displacement of lineages. These are US and famine era outbreaks. They're sharing, this is structure analysis of the microsat alleles, and these are two samples from Colombia and Costa Rica sharing structure with the US 19th century outbreak. Modern Mexican samples and modern US samples. Mexico's been a source of modern late blight outbreaks. Tomatoes and potatoes move across the border, and you see a lot of shared, this is all one cluster, Central America, Mexico. And we've done some PCA analysis as well of this. So interesting, the tools now are allowing us to track outbreaks, figure out where things came from, and we're figuring out how they migrate. These same kinds of tools could be used pretty much with any emerging disease, and we can go back into historic records. I've also, spent time doing work in the developing world. The Gates Foundation had a meeting called the ARC Agriculture Research Connections meeting, and while I was at AID, I did a bit of work with the Gates Foundation. I went to a grower's farm in Kenya, and this grower had late blight on tomatoes. This is a little dark, but this is Sigatoka on bananas. This is cassava virus on cassava. This is tree tomato. It's Solanum potassium, also a host of late blight. So this grower had a lot of the emerging diseases on that armed and dangerous list. So there's a big need for managing late blight and a lot of these other emerging pathogens in Africa. I've started a collaboration with Anne Nojirogi, Greg Forbes, and Jonathan Ewan. Greg's with SIP, and Jonathan's up in Sweden. And we're starting to track movement of Phytophthora infestans within Kenya and Uganda. This is some of Anne's data from her master species. Interesting, right now in Africa, they have the US1 lineage. It's present in both countries, but this new Kenyan 1 lineage when you do the microsat analysis on it, it matches up with lineages from Europe. And they're importing potatoes from the Netherlands and from the UK. You know, Kenya was a British colony. There's potato movement going into Kenya, and there's a concern that they'll introduce more diverse lineages and have more difficulty controlling this disease. So we also actually have historic herbarium samples from the first late blight reports in Africa, so we're going to dig into those as well to see. I suspect that most of them are in the 1950s, so probably going to be US ones. So how can we use this science to solve global food security issues? Well, USAID is funding work on transgenic potatoes. They're developing, a group at University of Wisconsin developed these RB, Solanum bulbocastanum potatoes that are resistant to late blight, and they're testing them in Indonesia, Bangladesh, and this is being run through the ABSC2 program out of Cornell. Short-term funding for scientists like Anne Nazarogi from SIP and from scientists from the developing world to come into laboratories in the U.S., either do their degrees and go back, or just short-term training studies are useful. We're running in-country diagnostic training, and doing more work with SIP in Africa. We'd like to see a global disease alert and mapping system developed for this disease. And also we're trying to use information and communication technology to track outbreaks. I'm developing some techniques right now that can be used on cell phones to run PCRs and to upload data into cloud computing sources, because then you can do some pretty low-tech in-field diagnostics and you don't need the expensive sequencing. And we're building collaborations through some of the programs. I've actually tried to get some funding from this Borlaug Higher Education Be Heard student program, and we're still working on some of that. So there's lots of new tools that can be developed to manage emerging diseases. This publication from the Chicago Council talks about fully deploying technology and tools to energize agriculture research in general, and also emerging disease biology, including bioinformatics, GIS systems, precision ag, molecular breeding, irrigation technology, biofortification. These are all tools that are really needed in the developing world, not just for management of late blight, but for just increased food production. As Fred mentioned, I spent a year in Washington as a Jefferson Science Fellow. This was my cohort. We had a nice photo op with the secretary. That was it, though. That was our one opportunity to meet the secretary. And Rajiv Shah was the head of USAID when we were there. And the group that I worked with was in the Bureau for Food Security and Ag Research Programs Office. And they have quite a big portfolio of work going on plant diseases. This is BFS, and it's, I don't know, the org chart may be different, Fred. You may know it differently from having been there. It kind of sometimes changes, but this is the group that we were in. But AID is a huge organization. It used to be in the State Department, and it's still a part of the State Department, but in a different building. And they have different bureaus: the Africa Bureau, Eurasia Bureau, Asia, Middle East, Caribbean. They're kind of, the bureaus are divided regionally, and then they have global health issues, a conflict bureau that does humanitarian aid, foreign assistance, the legislative group that deals with the folks on Capitol Hill to keep the money flowing. So it's a very big organization, but a very dynamic group, lots of people coming and going, foreign service officers that are deployed. It's like the military. They're deployed for a few years in country, and then they come back into Washington and work for a few years. And, we worked as part of this Feed the Future program. And Feed the Future has a lot of different goals, but supporting research and development to improve smallholder agriculture in a changing climate is really an important goal of what they do. And the plant diseases do affect food security, as I mentioned earlier. So they work in this Feed the Future countries are this part of Africa, across this region, East Africa, and also in this region in India and Nepal and Bangladesh. And then also, this is not the entire map, but there's also Honduras, Guatemala, and Ecuador and Central America. And they're working with different cropping systems, mixed maize in East Africa, and some other crops in other parts of the world, cowpea, and a lot of rice work going on in India and Southeast Asia, and also potato work. This diagram was was kind of developed at the time I was there. I'm not sure if they're still using it, but they have programs for climate resilient cereals, programs for advanced research on plant and animal disease, programs for legume productivity, and then it's kind of all underlying sustainable intensification. We've got the policy group working to make policies conducive for agriculture development. They've got people working on nutrition and aspects on aflatoxins and how it affects food safety. And then the HICD, the human capacity building component. So this is part of the research funding structure. And BFS had about $120 million budget. And so this combating plant diseases and pests of animals and plants is a big part of what they do. You know, they work on many of these pathogens that I mentioned earlier. They also work on East Coast fever vaccine development, and they have a number of projects being funded out of the animals group as well in the BFS. And they're interested in research along the value chain, so it's not just crop protection and plant diseases, but agronomy, breeding, technology adoption, and then harvest practices, how food is stored, how grain is stored can affect the incidence of aflatoxins, reducing the risk of affected, post-harvest handling, mismanagement of types of food harvesting, so trying to improve handling, and then also utilization research and kind of developing new ways to market and sell or even store crops through drying technology, solar drying, that type of thing. So they're looking at the whole value chain, and biotechnology is a huge part of the portfolio. They're working I have this slide that I borrowed from the group, and it just shows kinds of things they're interested in, developing tolerance to higher temperatures, increased photosynthesis efficiency, biofortification in cereals, reducing tillage through biotech-enabled weed management, increased water use efficiency, nitrogen use efficiency, tolerance. This is all things that we're interested in U.S. agriculture, also very important in the developing world. So they're funding a number of different projects, mostly a lot of the drought-tolerant work and nitrogen use efficiency is on cereals and work on corn. And they're using genetic engineering to address constraints in conventional breeding, and they also support late-stage R&D technologies that are suitable for smallholders. They're working very hard to transfer the intellectual property to public sector and local private sector groups within the Feed the Future countries, and they're trying to work to improve the regulation of genetically engineered crop plants. Right now, GE crop plants, there's several transgenic crop plant projects going on, cassava research on the cassava viruses, insect-resistant cowpeas, and then transgenic bananas with resistance to wilt, and they're working also on sigatoka. And this is kind of just shows some of the biotechnology projects, where they are in the Feed the Future countries, and who the players are, what agency, what groups are working. Many of the projects are being run through the ABSB2 project out of Cornell, but also with the International Rice Research Institute, IRIS, doing some of the rice biofortification. I mentioned SIP and IITA is at the Becca Hub in East Africa, and there are transgenic trials going on in Uganda. They have approval to run the banana transgenic work is happening in Uganda, but it's kind of country by country getting through the regulatory process, and they have a whole policy, a group there in BFS that works on this area, and then also at the State Department. The State Department has a group working on biotechnology transfer. In terms of late blight, transgenic potatoes are being tested in Bangladesh, and this is some data that Bill Fry shared with me of different areas in Bangladesh, and they're able to grow some of the transgenic RB potatoes with either one fungicide. The conventional potatoes without fungicides, you get a lot of disease, but they can really reduce the number of sprays. They aren't completely immune to disease, and they're concerned there might be overcoming of these transgenics genes, so they're pyramiding the genes now at SIP, but they do have to spray, but not as much as they would spray a non-transgenic potato. They're also doing a lot of capacity development, and BFS is funding the award program through the Gates Foundation, African Women in Ag Research and Development. It provides mentoring for women, African women scientists that want to continue in research, and then the Be Heard program is actually fully funded assistantships that's run out of Michigan State. This program, I think, is run out of Virginia Tech. But the idea is to increase capacity within countries so that the science can be done within countries in partnership with developed country researchers and develop, improve capacity to do agriculture research. While I was at AID, I went to the Gates Foundation's Grand Challenge recipients meeting to talk about the portfolio, and then I participated in this ARC program meeting on emerging agriculture research leaders. It's called the PEARLS program, run by the Gates Foundation, and they picked about 60, 70 scientists, African scientists, developed country scientists, and there's partnering on grant proposals, and they fund research. And this is the award group at the Becca Hub in Kenya that I spoke with. Also, while I was in Washington, I worked on the PEER Science Program. PEER is a program, Partnership for Enhanced Engagement in Research. This is federal government, U.S. government funded. And the years I was there, pretty much you had to partner either with NIH or an NSF grant recipient to write a PEER grant. The PEER grants are written by the developing country scientists, but you had to have a PI on the grant that had one of these two grants, either from NSF, from one of these agencies, NSF or NIH. I spent a lot of time going back and forth between USDA and USAID, talking with Sunny Ramaswamy and the folks at AID about broadening this out. They were already starting to partner with NASA, but they weren't partnering with USDA. And now, this year, success, it took 3 years, but now they're actually, you can, if you have a USDA grant, you can partner do a PEER Science grant with a developing country scientist. They are restricted by country, but it's not just Feed the Future countries, it's more expansive. If you go to the website, you can look, and so if you have collaborators you're working with and internationally, the money goes mostly to the developing country, but it's just a couple $100,000, so it's pretty good money for the developing country scientists to partner and to come to the U.S. and work on skills development. And so this is really good that they've expanded the partnerships now, because USDA actually is really one of the better fits for some of these development projects that are involving food production. I also was funded by the Bellagio Center and Rockefeller Foundation to run this Ecosystem Services and Emerging Disease meeting, and also from the Tech Futures Initiative. So we're working quite a bit now to try and build networks of people working on emerging plant diseases. And then I've been doing USAID workshops on disease diagnostics. I ran one in Honduras, one in Costa Rica, and I'm planning one for Africa next year at the Becca Hub.